Hey everybody, so for my URA project, I worked on a gene data set from some purple sea urchin juveniles. So, to get everybody on the same page about what we're talking about here, the purple sea urchin is a round-bodied, spiny, marine invertebrate, about the size of the palm of your hand, and as you may have guessed, purple in color. Urchins are an important species ecologically, as they can play a role in controlling algae populations. Additionally, in California, as a result of global warming, kelp forests, a huge source of marine biodiversity, are threatened partially by purple sea urchin foraging. Sea urchin embryos turn into free-swimming larvae called pluteus, and in turn, these larvae eventually settle on a substrate and become juvenile sea urchins. Once matured, the juveniles will have grown enough to become mature adults. This metamorphosis process is depicted in the middle graphic. As you can see, there is a juvenile stage, which occurs once the urchin has settled on a substrate, but before maturation. This juvenile life stage and the associated metamorphosis process from being a larvae is poorly understood from both a molecular and genetic perspective. To better understand juveniles, we can investigate their genetic response to environmental variability. By investigating how certain genes are differentially expressed in juveniles in response to different environmental variables, we can understand the relative importance of different physiological processes during sea urchin development. In our experiment, the substrate the sea urchin developed on was different between the experimental groups. So, by looking at which genes are most highly expressed in response to these more or less stressful substrates, it will indicate to us which physiological processes are most critical in sea urchin responses to stress during their juvenile life stage. To see which genes are most highly expressed in response to each of these set of substrates, sea urchin tissue samples were sent to an outside agency for Illumina RNA sequencing. We're given back data files which contain raw RNA read data, which we then prepare for gene expression analysis. We filtered the read data for quality, removing probable error regions, and then the reads are assembled into a transcriptome using a program called Trinity. Several other programs integrated into Trinity then allow us to check the assembly's quality and align the reads to the genome and perform annotations in preparation for differential gene expression analysis. Differential gene analysis is where the relative expression level of relevant genes are compared, and based on this comparison, researchers can identify whether a particular gene has a specific function in juvenile development, or if it's even important at the juvenile life stage. My role in this research project concluded at this stage by handing off the prepared RNA dataset to other members of my lab for further comparative gene expression analysis.